everyone, I welcome you all to my presentation today. In this presentation, I'll be talking about our study on CpG-ODN increased antimicrobial immunity and cellular metabolism in broiler chickens. Let's start with a little bit of background. There are a number of pathogens causing diseases in chickens such as bacteria, virus, fungus, and parasites. I'm going to focus on bacteria here as most of the bacterial diseases increase the mortality and decrease the quality and the quantity of the final product. Nowadays, usage of antibiotics has become questionable because of increasing antibiotic resistance. Therefore, developing alternatives is very important for the benefit of poultry industry. CpG-ODN or cytosine phosphodiester guanine oligodeoxynucleotide, which is a short single-stranded synthetic DNA molecule. This is the molecule we are focusing in our study, and you can see the nucleotide sequence here. It contains cytosine, guanine nucleotides, and also 2-deoxyribose sugar, and in between there is a phosphodiester linkage. This CpG-ODN represent CpG motifs of bacterial DNA, and it can stimulate the immune system of vertebrate animals, including chickens, without causing any diseases. How CpG-ODN mediates its cell signaling? As I mentioned in the previous slide, it represents bacterial DNA, so it acts as a pathogen-associated molecular pattern, so PAMP, which is recognized by pattern recognition receptors. TLR9 in mammals and other species, but in chickens it's TLR21, present in endosomal membranes of antigen-presenting cells. Upon this recognition, it can stimulate the intracellular signaling cascade and gives signals to the nucleus to produce more inflammatory cytokines such as interferons, interleukins, TNF-alpha, which are responsible to recruit the other immune cells to the site of infection and to fight against invading pathogens. Studies conducted in our lab could prove that CpG-ODN can protect chickens against E. coli and Salmonella bacterial infections. Here you can see increased survivability of CpG treated group compared to the saline control. And further, previous studies confirmed this effect using different delivery routes: intramuscular, subcutaneous, in ovo, and intrapulmonary routes. Moreover, a recent study conducted in our lab revealed that it can change metabolic profiles of chickens following CpG-ODN administration. Some metabolites like amino acid and purines decreases while some increase like 2-hydroxybutyrate, 3-hydroxybutyrate and formic acid, and mild increase in TCA cycle intermediates. So the major antimicrobial activities of innate immune cells in a chicken immune system we can recognize as phagocytosis, oxidative burst, degranulation and nitric oxide production. Phagocytosis we can mainly see in macrophages, granulocytes and dendritic cells, and oxidative burst, heterophils and macrophages are responsible for that. Degranulation mainly happens in granulocytes. So in our study, we focus on these three major mechanisms. And cellular energy metabolism regulates immune cell fates and function. We can highlight glycolysis, Krebs cycle, and electron transport chain as the key energy producing pathways in an immune cell. These pathways changes when there's a immune cell get activate and it undergoes profound metabolic changes to meet energy demand and cellular biosynthesis and facilitate the signaling processes. As the immune system consisted of heterogeneous immune cell population, different types of immune cells prefer different metabolic pathways to produce energy when there's a energy demand. So the considering informations from previous studies and other background informations to increase the utility of CpG-ODN as an alternative to antibiotics in chickens, we need profound understanding of its mechanisms. As informations are lack on this basis, we plan to start from the front line of defense in chickens. So our objective was to investigate the antimicrobial mechanisms and associated cellular metabolic pathways of chicken immune cells after CpG-ODN administration. So this was our experimental design. We had two groups of 2 to 3 week old broiler chickens, and one group was injected with CpG-ODN at the dose rate of 50 microgram per bird intramuscular route, and other group was injected saline as the control group, and blood samples were collected after 48 hours of injections, and the samples were processed to measure the antimicrobial activities and metabolic pathways of immune cells. Immune cells were isolated and measured the antimicrobial mechanisms. Phagocytosis using Phago test kit, oxidative burst using a Phago burst kit, and degranulation we used anti-LAMP1 antibody to stain the degranulated cells. And here I want to mention about these two kits. These were originally designed for mammalian immune cells. Therefore, we had to optimize almost all steps, and we optimize a new protocol to measure chicken immune cell functions, and the results were obtained using flow cytometer. To measure the metabolic pathways, cellular glycolysis and mitochondrial respiration were quantified using real-time novel technique using Seahorse XFP analyzer. Here, glycolytic rate of the cells measured using extracellular acidification rate, and the respiration of mitochondria are measured as oxygen consumption rate of these cells. Here are the results of our study, phagocytosis results, and this graph shows how we separated heterophile population using scatter plot and specific antibodies. And according to this graph, you can see the results of phagocytosis percentage, and the phagocytosis percentage of CpG given group is around 38%, and in the saline control it is around 20%, and this difference was statistically significant. Oxidative burst results, we found that heterophiles burst effectively around 60% of burst activity when incubated for 5 minutes, and when we increase the incubation time up to 10 minutes and 30 minutes, the ability of heterophiles oxidative burst activity decreases, and in this graph you can see the percentage of oxidative burst, and this was significantly higher in CpG treated group compared to the saline group. In degranulation results, we saw the same pattern, and CpG treated group had significantly increased degranulation ability compared to the saline group. Results of cellular glycolysis. In this graph, you can see how extracellular acidification rates change with the time. Once we inject glucose and PMA, glycolysis rate increases. After injecting rotenone, antimycin A, and 2-GG, it decreases. So by adding these stimulants and inhibitors, we can measure the rate of glycolysis in isolated cells. Red line showed CpG treated group, and blue line is the saline control, and there is a distinct difference between these two groups. Then we calculated compensatory glycolysis. As you can see here, CpG treated group expressed higher compensatory glycolysis, and this was significant compared to the saline group. Mitochondrial respiration. In this graph, x-axis represents oxygen consumption rate and how it changed with the time. And the oxygen consumption rate was always higher in CpG group, represent red line, and compared to the saline group. Spare respiratory capacity, as you can see in this graph, it was higher in CpG group compared to the saline group, and this spare respiratory capacity We can describe it as the amount of extra ATP that can be produced by a cell by using oxidative phosphorylation pathway in case of sudden energy demand. In discussion and conclusion, based on the results of our study, we can say that CpG-ODN significantly stimulate the antimicrobial activities, oxidative burst, phagocytosis, and degranulation of heterophiles in chickens, and this is likely the major mechanism of CpG-ODN mediated protection in the chickens experimentally injected with lethal doses of different bacteria, E. coli and Salmonella. And also CpG significantly increase the immune cell activation and metabolism, both cellular glycolysis and mitochondrial respiration. With that, I am going to end my presentation, and I would like to thank my supervisor, Dr. Susantha Gomes, for his continuous guidance and support, and my advisory committee, Gomes Research Group, for their help and support for conducting trials, samplings, and sample processing throughout these experiments, and University of Saskatchewan Western College of Veterinary Medicine Animal Care Unit, Clinical Pathology Laboratory. Thank you very much, everyone. And our funding sources: University of Saskatchewan Graduate Scholarship, NSERC, Canadian Poultry Research Council, Chicken Farmers of Saskatchewan, Western Economic Diversification of Canada. Thank you very much all. If you have any questions, I am happy to answer, and your comments and suggestions are also welcome. Thank you.
Hello, Iresha. Congratulations on your presentation.
Thank you.
It was very interesting, and also it brought a very interesting explanation to the reason why CpGs, when added to certain proteins, will improve their efficacy in whether protection or immune response. So that's something that has been it's been very I would say insightful to hear your presentation in that regard. There is a question from the floor, and the question is the following: great work. Maybe I missed this, but so you measured heterophile numbers in tissues to check the phagocytosis, or did you do in vivo studies using cultured heterophiles and infected E. coli to measure the phagocytosis? If you want to look at the question, you can open the Q&A box at the bottom. It's often easier than hearing my accent.
Yeah, actually, to answer this question, we collected samples from birds, actually, like from organs and tissue samples. Especially here we focus on the blood samples, and then we did the in vitro study. Like it's kind of a mixture of like an in vitro study and in the chicken. So because we collected samples from the blood, and then we measure the functions in the lab by adding E. coli, which is labeled with fluorescent, like FITC labeled E. coli, and then we kind of stimulated cells for around 30 minutes, and then we measured the phagocytosis. So that's how we did it.
Okay, well, thank you very much.